This is a newcomer Streptococcus bacteria, which is a pathogen that just coming into the respiratory tract. Now, as he have entered the respiratory tract, he start to ask one of the experienced pathogen about the spikes on the petrolling cell, which is the B cell. The experienced pathogen then warned the newcomers about how he could be killed by the B cells through neutralization and agglutination process. For neutralization, antibodies will bind to the antigen of the pathogen, blocking the pathogen from entering the host cells. This will prevent from initiate a new infection. For agglutination, the binding of antigen and antibodies will cause a large complex, which is a clumps, and it will be eliminated. Bye-bye! Here we can see there are some cells are patrolling the front doors of pathogen interior cells. These pathogen need to pass the patrolling cells in order to cause infection. One of them are helper T cells. Helper T cells will firstly identify the newcomer, either it have their target or not. Unfortunately, the pathogen are their target. Therefore, helper T cells will call all the B cells and instruct them to attack and exterminate this pathogen.
Hello, good morning students. So for today's lecture, I will focus on B lymphocytes. There will be no slides as it will be a very short lecture. But don't worry too much because I will share a few diagrams and some animation for your better understanding on this topic. Before I forgot, we will also have some quizzes at the end of this lecture. So I expect you guys to pay full attention on what I am going to share and get high marks for the quizzes as it will contribute around 50% for test 2. At the end of this lecture, you should be able to understand the activation and clonal selection of B cells, identify the structure of antibodies, and also explain how antibodies react to antigens. So, there are 3 learning outcomes in total. So the first part of my lecture would be activation and clonal selection of B cells. It's B cells, not beta cells, okay? B cells, also known as B lymphocytes, is actually a type of lymphocytes. Let me share with you a very simple diagram. The activation of B cells begins when the B cell receptor binds to either soluble antigen or membrane-bound antigen. The activated B cells will then undergo clonal selection, which form clones of B cells that possess the same antigen-specific receptors. Then, the clone B cells will differentiate into plasma cells or memory B cells. Plasma cells will secrete specific antibodies that circulate in lymph and blood to bind to free antigen. Meanwhile, memory B cells will provide immunological memory and quickly react to antigen to stop infection. Group 4 will explain further on plasma cells and the reactions. So moving on to the next part of my lecture, which is the structure of antibodies. So this is the structure of an antibody. Antibodies are glycoproteins that belongs to the immunoglobulin superfamily, consisting of 2 heavy polypeptide chains and 2 light polypeptide chains. There is a variable region at the tips of heavy and light chain, which is composed of a bivalent antigen binding sites, while the rest composed of a constant region which will determine the mechanism used to destroy antigen. The constant region is called C region. Okay, so here is the last part of my lecture, which is the antigen and antibodies interaction. One of the antigens and antibody interaction is neutralization. Okay, so here you can see the comparison between normal viral infections and viral infections when neutralization takes place. During the neutralization, the antibodies bound to antigens on the surface of an infectious particles and block its ability to bind to a host cell, and thus preventing it from destroying the body cells. Okay, next diagram shows another antibody's action, which is agglutination. Firstly, the antibodies called agglutinins will bind to the antigens. Then, it will cross-link the antigens to one another, creating a large complex. The large clumps consisting antigens can then be easily eliminated by phagocytosis. Okay, that marks the end of my lecture. So now let's answer some quizzes. Thank you.